Hi everybody, welcome to session 10. Today we're going to be talking about B-cell activation and humoral immunity. This is based mostly on chapter 10 in your textbook, so remember to try to have your textbook with you so you can follow along in the figures, as they might be a little bit easier to see in your book than on the screen. So for today's lecture, we're going to talk about B-cells activated first by T-dependent antigen, meaning they require help from helper T cells. We're going to talk about how those cells together form something called a germinal center, and affinity maturation of the antibody, meaning that the antibody is going to actually increase in its affinity for its antigen through a process called somatic hypermutation. Next, we're going to talk about how the antibodies are produced, and then also the goal of this is to generate two different types of mature B cells. A memory B cell, so that if you were ever to see the same antigen again, you'll make a stronger memory response to it, and also to make plasma B cells. These cells become antibody factories to help fight the current infection. Then we're going to switch and talk about B-cell activation by T-independent antigens. So for these antigens, no T-cell help is required. And lastly, we'll spend a little bit of time talking about what all the different antibodies that are made by the B-cells actually do. So you'll learn more about this in subsequent lectures, but I'm just going to give you a little bit of an introduction about how the different types of antibodies have some different functions and different locations within the body. So we're going to start off talking about T-dependent B-cell activation by antigen. The first thing that happens is that the B-cells actually have to see their antigen, and they're going to do that through their B-cell receptor, and that's going to occur in a lymphoid organ such as the spleen or a lymph node. Then they actually have to interact with the T follicular helper cells that are going to help them to produce more antibody. This linked recognition of antigen by T cells and B cells is really important to get a stronger, more robust antibody response. Remember that B cells and T cells are normally found in two different locations in lymphoid organs. So once B cells see their antigen, they actually have to move so they can be closer to the T cells. We'll talk about how that process actually occurs. We'll also talk about surface molecules as well as secreted molecules such as cytokines that help to promote this process. Then we'll talk about how the B cells are activated and differentiate into, again, either plasma cells or memory B cells. The second phase of B cell activation by T cells occurs with the formation of a germinal center. In germinal centers, the B cell receptor or antibody will actually undergo somatic hypermutation. So remember in a previous lecture you learned about VDJ recombination and how that gave specificity for one particular antigen. Now that B cell is going to try to perfect that antibody so that it can bind even more strongly to that one particular antigen. And it does this through a mutation process. There will be positive selection for the B cells that have antibodies that mutate where the binding actually improves, because think that some mutations might result in an antibody with a decreased affinity, and those B cells will actually simply die off. This process is mediated by an enzyme called activation-induced cytidine deaminase, abbreviated AID. That is actually going to facilitate the mutations, and we'll talk about how that happens. Now, with these mutations, the cell is going to recognize the mutation. So different repair enzymes will come into play, and depending on which repair pathway is activated, different types of mutations will end up being created in the genes that are going to make the B-cell receptor or antibody molecules. AID also initiates class switching. Remember when you learned about VDJ recombination, that's for the variable region of the antibody. But there's also a constant region, the sort of bottom part of the Y. And different constant regions for different types of antibodies, whether it's IgM or IgG, IgA, those different constant regions have to get switched in to bind to the same variable region on the antibody. And AID is also going to facilitate that process. Cytokines made by the follicular helper T cells are going to direct the B cell for which class of antibody it should make. And then, of course, ultimately the goal through this whole process is that we're going to have a B cell that makes an antibody with really high affinity for one particular antigen. And once it's done that, it's going to go out into the periphery as a plasma cell to make lots of antibody to help you fight an infection. And a subset of those B cells will become memory B cells and move back to the bone marrow and wait until they're needed again in case you see that same infection again later on. This slide is just a brief overview of activation by T-dependent or T-independent antigens. And again, we're going to focus on T-dependent antigens to begin with. What's important to note here is that a signal comes from the B-cell receptor, which you can see, and then a second signal is coming from MHC2 interaction with the T-cell receptor. For two T-independent antigens, which we will discuss later in the lecture, you can see there's no T cell, and both signals are actually coming from the B cell itself. So here's an overview of how T follicular helper cells respond to the antigen presented by a B cell and what that results in. So again, you see down below the B cell interacting with the T cell through its MHC class II and the T cell receptor on the follicular helper cell. You also see CD40 and CD40 ligand. That interaction between the B cell and the T cell is very important for antibody production. Cytokines are also produced by the T cell. The first thing that happens is that will induce the B cell to proliferate. Then it will ultimately go on to form a germinal center, and then again those plasma cells or memory B cells. So we're going to break down the processes that occur at each one of these steps. A key point to keep in mind through this lecture of T-dependent antibody production is how there's linked recognition by both the T cell and the B cell for antigen. But remember, T cells and B cells see antigen very differently. T cells only see antigen presented on MHC molecules, so they see small peptides. B cells see antigen in its native form. For instance, here you have a whole viral particle, and the B cell receptor is binding to an antigen on the surface of the B cell. But remember, that will induce the B cell to engulf that virus, and once inside, it will chew it up into pieces, and it can present many different potential pieces on MHC to a T cell. Also keep in mind, though, that the T cell may see antigen presented by other antigen-presenting cells, such as a dendritic cell. Now, where do B cells see antigen? If you think about how our immune response is initiated, normally we have innate cells at a barrier site, such as the skin or in your lungs or a mucosal site. They're at sites in your body where you're likely to have a foreign antigen come in. But that's not where B cells are. B cells live in the spleen and live in the lymph node. So how does B cell see antigen? The antigens have to come through the blood. Now, if you have a blood-borne pathogen like malaria, the antigens will be in the blood. But remember, a lot of infections don't like to be in the blood. So the antigens have to still get transported through the blood, but they'll do that on innate cells, and we'll talk about that on the next slide. But once they come into the spleen, then the B cells can see what those antigens are, and that happens in these follicles. So, antigen transport into lymph node and spleens. There are two innate immune cell subtypes, which are subcapsular macrophages and also follicular dendritic cells. Both of these cell types are very good at binding antigen, but they actually don't internalize the antigen. So they don't pull it inside and break it up into pieces and present on class 2. They simply keep the antigen on the surface, which you can see here. Now that's important for B cells because when those cells come into the lymph node or the spleen, the B cells can then sample what's on the surface of these cells. And the B cell then can take that antigen off of those cells, internalize it itself, and present pieces of it on class 2. During this process, the B cell also gets some survival signals from these antigen-presenting cells. It's the first survival signal the B cell will get because it hasn't had a chance yet to get that signal from a T cell because it hasn't interacted with a T cell yet. Remember, it's still in the follicle, and a T cell is separate in the T cell zone. So that's the next step in the process. After B cells encounter their antigen, they go looking for T cell help, which means they have to move from their follicle closer to where T cells would be in the spleen. Now, if B cells and T cells are separate in the spleen, something is keeping them tethered there. And that's actually chemokines and chemokine receptors. So B cells express CXCR5, and that keeps them in a follicle. And T cells express CCR7, which keeps them in the T cell zone. So what happens is that each of those cell types actually stops expressing the first chemokine and starts expressing the chemokine that the other cell makes that allows them to move closer together. So B cells start making CCR7 and T cells make CXCR5. That allows the cells to move to the border of each of their individual locations and allows them to interact. Once they interact, then both the cells still have to move into what we'll learn about as a germinal center. Now, the T cells and the B cells first interact when B cells present antigen on MHC class 2 to the T cell receptor. And then the T cell will interact with the B cell through CD40 and CD40 ligand. But that's not enough to hold those cells together. So both B cells and T cells express integrins that are called SLAMs for signal lymphocyte activation markers. The B cells are always expressing these, and the T cells are as well, but these two integrins can only stick together when there's another helper protein called SAP for SLAM activating molecule. So once a T cell sees antigen, it will upregulate SAP, and then this allows the T cell and the B cell to stick together very well because they need to interact for a long time in order for the B cell to really get the help from the T cell that it needs. So I want to just spend a little bit of time on this table and talk about the different terms for the different B cells at their different activation stages. So first off, you have a resting B cell. This is a fully mature B cell that's come from the bone marrow, but it hasn't seen its antigen yet, so it's naive. It has a lot of surface Ig or B cell receptor. It also has a lot of MHC class II, so it's ready to respond to its antigen, but it hasn't seen it yet. It doesn't secrete any antibody yet because it hasn't been activated. Once this resting B cell sees antigen and gets activated, it becomes a plasmablast. Now, that's not the same as a plasma cell. So a plasmablast is an early form of an activated B cell that can make antibody. Still has a lot of surface IgG and MHC class II, but now it can secrete antibody. Eventually, through the maturation process that we're going to discuss, it becomes a plasma cell. It actually no longer has a lot of antibody on its surface. It doesn't need to interact with antigen anymore, but it does need to make a lot of antibody. Now, a resting B cell has the ability to grow, make many more copies of itself, go through this mutation process to improve its antibody affinity, and also class switch. A plasmablast can do many of the same things, although it's unknown how much a plasmablast is still mutating to improve its affinity. What is definitely known is once it becomes a plasma cell, it doesn't need to make more cells, it just needs to make antibodies, so it stops dividing. It doesn't need to increase its affinity anymore, it's already done that, so it doesn't mutate anymore. And it's already class switched, so it doesn't need to do that again. All right, the second phase of primary B cell immune responses to T-independent antigens, again, occurs in lymph nodes and spleen, and it involves the formation of a germinal center. So when you look at this picture here of a lymph node, again, the yellow is where you would have B cells. The blue is where you would have T cells. Once the B cells and the T cells move, each to their border, you start to get this darker yellow circle. Eventually, that is going to become a germinal center. Now, germinal centers are about 90% B cells, but there have to be some T cells there to give them help. There also have to be some of those follicular dendritic cells to feed them antigen. And a germinal center doesn't last forever because you only want it when the antigen is present in your body. So usually they exist for a few weeks and then they start to go away. And that's what's shown here with this smaller circle, that the germinal center is actually disappearing because it's no longer needed. Here's a close-up of a germinal center. Now, the cells in yellow are mostly your B cells. In blue are helper T cells, but the bigger blue cells here in the center are your follicular helper T cells. So, you also have two zones in a germinal center called a light zone and a dark zone. That's about how they look under the microscope. It doesn't really tell you much about what happens in each place. B cells in the light zone will still interact with those follicular dendritic cells, as shown here, this cell in red. So they're still pulling antigen